Start our class. There aren't very many people here, and but we're going to get going and see what happens in the next couple minutes. So here we are. The last Thursday was a snow day, and I recorded a lecture, sent a link out to that. You don't send me the notes on that because no one was in class. But it's as important as any other class meeting, and so you really need to watch it and watch it. Hopefully, you've already watched it, because we had canceled class, we're not going to do that clay assignment. We'll move ahead to another assignment we'll do in class. We'll talk about that. Then Riley was going to visit today and got hung up. He works in the emergency department and worked all night. So he's going to be here on Thursday, and he's going to be our CAR T cell therapy person that we've talked about through the semester, and also our tutor, and our special, material tutor. He knows his stuff. He took the class last spring. Where we are this week is you should have read or be reading chapter 4, and then we're going to move on from there to chapter 9. So the order we're reading the chapters are 1, 4, 9, 17. And the 17 is the first of 4 chapters in the 4th section of our book. And the 4th section, I think, is called Cell Regulation. We could call it cancer. The whole thing is focused on cancer biology. So we're pointing at chapter 17, and we've been pointing at chapter 17 right from the first day. And we're building up to it, right? Because, that's the last section of this book, so it must be some of the more challenging material. And our whole course is focused on getting to that. And when we do, after we do the 4th section, we'll loop back and we'll pick up chapters from sections 1, 2, and 3 that we haven't covered yet. And so that's the flow, okay? And so where we're at is today we're going to start getting into some of chapter 4. And let's see if I have any slides about chapter 4. So chapter 4 is really about these clay model videos. Chapter 4 is the foundation of molecular biology, and molecular biology is all about gene expression. And so this is all hardcore molecular biology. So when you start looking at chapter 4, it talks about genes and chromosomes, identification of DNA, genetic material structure, DNA replication, role of mRNA, genetic code, blah, 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 like that, And so we'll talk some about chapter 4, but chapter 4 is another introductory chapter, like chapter 1, and I don't really feel the need to talk through it because it's a mixture of some intro bio review and then some really well-described stuff. And instead of following along exactly with the book, what we're going to do is we're going to pick up an example that's going to head us toward section 4. And we can use that example, and it's the example of our author's career. We can use that example to roll through every aspect of chapter 4, and we can use it as an example. So when you have questions for chapter 4, I say, this is how we could explain that method, is how the scientists used to study this topic, like that. And so that's how we're going to flow through chapter 4. Then chapter 9 is about transcription regulation, and the author talks like a lot of authors do. The author covers a lot of territory in it and doesn't really create a good just framework for the concepts and principles of the chapter, which that is, right? So we're going to roll through chapter 9 and we're going to pick up pieces of it without losing this foundational stuff. And I have your quiz back. I graded that, and I'll hand that back into the class. I should mention up front here, I made a 10-minute answer key video, and I'll send the link out to that when I send out the link for class today. And so at the end of class, when you get your quiz back, you'll be like, this is marked wrong, but I don't know why it's wrong. So the first thing to do is to watch the answer key video, because I try to explain things in that, and then we want to follow up on that and learn about that, because it's this foundational stuff that's going to drive us through these chapters as our core example and our really most important concepts, okay? So that's the flow of stuff this week. I should stop and check in to see if anybody has any questions about anything at all. And so remember, the first time I taught this class, it was just, I'll answer your questions like that. And this is different because that's pretty challenging. But questions are really great. And your questions can range from very specific things, maybe from the snow day lecture, maybe from the first day of class, maybe from a specific figure in the book and stuff like that. There were even some questions about from the book, what do we need to know from the book, and that's a really good thing to talk about. And so I bring my book into class so that you can just say that. You say, oh, I'm looking at chapter 462, and you don't have to have sophisticated questions about it. You can just say, I'm looking at that, and we can all turn our books to that, right? There should be a lot of that. So please keep track of that as you read and write down questions, or just like a page, right? Like that. That's really good. Okay, so this week you have to decide if you're going to do the 4-H STEM Ambassador Program or you're going to do a term paper, which is toward the end of the semester. And if you do the 4-H STEM Ambassador Program, you just contact Sarah Sparks like that. And she helps you organize it like that. This program was completely shut down in the pandemic, so there's just a few students that are going to be doing it this semester like that, still firing it back up. But you have a question?
This is back to when Riley comes in. How much of that is going to be tested on? Come up
All of
with
it, yeah. We'll use Riley's stuff.
Okay.
Riley will be talking about his CAR T-cell therapy, which is only described in one figure in chapter 20. So there isn't very much about CAR T-cell therapy in our book. But like I said, Riley took this course and knows what's taught in this course. And I'm meeting with Riley, and he's going to be trying to teach it to you in a way that fits right in, as an example, like that. So we have this example of clay model videos. We're starting to have an example from this snow video of what happens to our skin cells when they heal under a scab, and the signaling response and the cell response. And we'll loop back and talk more about that today, because that's going to be our second major example. And Riley's just rises out of that. I talked about some of Riley's stuff to start introducing it in the snow day lecture. Yep. Yeah, but it'll be just as important as other stuff. I picked the example, though, of CAR T-cell therapy because it fits in really well. It's like, well, we should be able to understand that really well. And it's pretty interesting because it is a challenging therapeutic strategy and to understand, but it'll take time. And Riley's going to visit class 4 times like that. Yep. And so this 4-H STEM Ambassador thing. And if you have any questions, you can ask me, but you can also contact Sarah Sparks. She's got all the answers. And it's a small program this year, but it's a really good experience, and super recommended if you could possibly do it like that. And again, if you have questions, there's just that training information. The training starts next week. That's why you got to decide this week, yes or no, you're going to do that or not. Okay, great. Okay, so then I finished talking about chapter 1 either last week, either in class on Tuesday or in the snow day lecture. And I fit those together, and I just want to mention that the way I fit those together is like this, where I say, before I do that, I want to say, and before we leave it behind, there are these 2 sections, and I talked about them some the way I wanted to talk about them, and then I talked very briefly about the 3rd section, and the 3rd section is about some methods, like, oh, look, we're using microscopes, or we're using centrifuges, and it's our first chapter. It's really nicely written, and there are some examples of methods that I'm not mentioning, but it's good to know in a 2-point way, like, well, what is centrifugation, and what are its uses? And there are some different kinds of centrifugation like that, and some different uses of it. And I described one really good use, and that is to create a nuclear extract. And I described that in the snow day lecture last week. And you use a Dunce homogenizer to lyse the cells but retain the nuclei, and then you use a centrifuge to spin the nuclei down and make a nuclear pellet. And you have the nucleus, a functional nucleus where genes are being expressed, but it's no longer inside a cell, like that. And so that's just a thing where you should ask questions. You should ask questions about this or that, the other thing, if you don't understand it. Because my thinking is, well, you read the book and you're like, oh, I can't understand that. And you don't have to understand everything, right? We're framing these as 2-point questions, right? So I'm not going to be like, tell me 10 things about that figure, right? It'll be like, well, give me an example of centrifugation and how it's used, like that. Right? It's hard. I think the exams are harder than our quiz, but like our quiz, where they're just a bunch of direct questions like that. What about this? What about that? And so that's the learning we're targeting. And so I'm not going to talk again about section 3 unless chapter 1 unless you bring in questions. And we can bring in questions anytime during the semester about chapter 1. I laced these together, talking about evolution and talking about model organism. And I just put them together because it's the center of our course. And it's the center, a huge center of biology, and molecular biology sits there and represents that. And the best way to talk about it is to do a blast search. And so in the snow day lecture, I just said, let's just do a blast search. Let's take human myogenin protein and look for it in a dog. And it's no problem for a computer to find a very, very similar gene in a dog. And scientists could study it and find, oh, it's myogenin. And the dog myogenin does the same thing. And the interpretation is this concept of an ortholog. And ortholog has a very strict definition. It's two genes in different species that share a common ancestral gene. And you just need to memorize that and be ready to say that. And then provide examples like myogenin in humans, myogenin in dogs. Myogenin in fruit flies, because I showed that. So the first thing is, wow. These little highlighted things that I talk about, these little highlighted things are just this remarkable similarity in different species with myogenin. So remarkable that you just couldn't explain that in any other way other than that there must have been a common ancestor. Because things just don't happen that way in our universe. This universe isn't big enough for those to be that similar by random chance, right? But the dog myogenin amino acid sequence and the human myogenin amino acid sequence are so remarkably similar that nothing in our universe has ever happened that way by chance. Just no way. Right? It's not a 1 in a million, 1 to the 10 to the minus 6 chance. It's a 1 in 10 to the minus 115 chance, right? It's just like that. And that's fine, great. It's not surprising that the human and dog are really similar. When they're that similar, that allows us on life on our planet to just look across just great expanses of evolutionary history. We can easily find the myogenin ortholog in a fruit fly, even though that last common ancestor that had that ortholog of myogenin lived like 600 or 700 million years ago, right? And you can, people do just kind of shrug and be like, yeah, 600 million years, but there's no way, we can conceptualize 600 million years of time. It's just a ridiculous amount of time. And then but I think this is the fruit fly one. And, fruit fly myogenin, human myogenin, and just looking at the sequence similarity, it's not 1 in a million, 10 to the minus 6, it's 1 in 10 to the minus 20th, that these would be that similar by chance. So because of this astonishing similarity in amino acid sequences, we can just range over great evolutionary distances. We can see relationships between a gene in a human and this appropriate gene in any species on our planet, like that, and then you can just do it for thousands of genes and map all their evolutionary relationships out, and that computers have no problem storing that information. Scientists have no problem collecting that information. The process is a method called DNA sequencing, where you read the letters of the gene sequences in any species, and then computers can make those comparisons very, very fast like that. Yep. So that's that, and that's that key concept. And that's the heart of the concept of a model organism. You say, what's fruit flies, why is that a model organism? The answer is, it's astonishingly closely related to humans. Like that. And you just don't see that. Well, you don't feel that. No person ever thought that or proposed that in the history of Earth, right? Leonardo da Vinci did not say, hey, I really think that fruit flies and humans could be shown to be astonishingly similar. No way. What they appear, right, as you're walking on a nature walk, they appear to be astonishingly different, right? Bones, no bones, wings, no wings, right? Like astonishingly different. People marvel at the diversity of life on Earth. Molecular biologists point toward the commonality. Evolution is like a two-sided coin, and one side is the commonality, and the other side is diversity of all life on Earth. And there's astonishing stuff on this commonality side, and that's what we would talk about in this kind of course. And so that's what I would want to do when I talk about evolution and model organism, introduce it. And I just think it's bizarre and ridiculous that that's not in there. To just to be like with a whole bunch of exclamation points. Like, look at this crazy data. Pick a gene, myogenic, whatever. Look at that crazy data of the similarity across these species. And again, the important concept of an ortholog is that we look at that sequence similarity, that provides one piece of evidence, and then we also need to look at the function in the organism. And so if we studied humans and dogs, we'd see that myogenic does the same thing. It's a transcription factor. It binds the enhancer of muscle-specific genes, and it activates them as the cells are differentiating into muscle cells, right? And the same thing in fruit flies, like that. And we don't study every species. We just say, hey, we've studied a handful of model organisms across a great evolutionary range, and so I bet it's the same for everything in between, right? All mammals, all vertebrates. Did T-Rex have a myogenic ortholog? We'll never know, but right? Yeah. Like a one that was really cool and had, big teeth, like that, stuff like that, all right? And that arises from this, which people don't talk enough about. They talk about evolution and people think it's a process, but it's a process and a pattern. And the thing that biologists are really excited about is not just the process, but the pattern. Everything in biology is about the pattern. You can't find an example in biology, in the story of life on Earth, that isn't a pattern that appears to be a product of the process of evolution, whether it's rapid evolution, like COVID variants, or really slow evolution, like the evolution of a horse's leg, or the evolution of a leg, like that. Yep. And so all this stuff is pattern, right? The concept of model organisms is based on the guarantor that evolution provides similarity, commonality down at that molecular level. So we're good with that. And I like to talk about those multicellular model organisms. The molecular biology has its roots back in the 1950s down at this really, really small scale level. Molecular biology is a reductionistic science, so the reductionists say, well, let's study humans by studying the simplest thing we can. And so they said bacteria, but then this shows these bacteriophage, presence of these bacteriophage, which are viruses that infect bacteria. And those were the first things that were studied. And there's still a lot of virology in molecular biology. Our author started his career as a virologist in Wisconsin. And you don't need to remember that he was in Wisconsin. I'll talk about some names just because a lot of these people are alive still. It's a young science and a lot of these people are old, but like Jim Watson, co-discoverer of the structure of DNA, he's still alive. Super old, but still alive. And so it's just good to talk about that because it can also take you to a place in your mind and you can add some learning there like that. So, we'll talk about Madison, Wisconsin, not because you need to know that for a test, but it'll help you remember what our author did in graduate school there, like that, okay? And so in that, we list these model organisms that molecular biologists really like to study stuff in labs under controlled environments, and they really like to pick on about a dozen or less than a dozen of these model organisms really heavily. And those include viruses and bacteria, and but not just any viruses. There's some viruses that are really picked on a lot like that. And a lot of us, this is taking these general things, but also adding, okay, but which viruses are ones that we should especially learn about? And so in the viruses, we're going to learn a lot about a certain type of virus, and we'll talk about it a lot, and it's the one type of virus that our author studied in Wisconsin. It's a really famous one in cancer biology, and they're called retroviruses. And just as an example, human HIV is an example of a retrovirus like that. And they're called retroviruses because the virus itself has RNA in it, and when it infects a cell, it first needs to copy that RNA into DNA. And that's called reverse transcription. We learned in the clay model videos that DNA gets transcribed into RNA, and that's how genes are expressed in every cell. A retrovirus has to actually infect a cell and have its RNA reverse transcribed into DNA as a first step. And we'll learn a little bit about the life cycle of that virus today because, again, it fits into the story of cancer biology as a major driver in molecular biology since 1970 when our author was in Wisconsin, like that. And so here's just a name of one, and this name is not that important, but it's just a name of one, just to show you that's a particular retrovirus. And the author and molecular biologists and cancer biologists are not interested in every retrovirus. There's a set of about 24 weirdo retroviruses that they study, starting back in the 70s when our author was in grad school. And these are weird variant retroviruses, not found in humans, but found in some species like mice and monkey and chicken and a couple others like that, the virus actually causes cancer, like that. There are some other viruses that are associated with cancer we'll learn about in chapter 20 when we really talk about cancer biology, but these retroviruses come up early, and they come up because there's some variants, again, not in humans, but some variants that were identified a long time ago that cause cancer upon infection. And way back in the 1970s, molecular biologists got a foot in the door on cancer biology, and they did it studying those retroviruses. So the scientists that studied that Rouse sarcoma virus in 1970, they got a Nobel Prize because it was nuts that they discovered how the virus was causing cancer. Our author did that work. Our author's entire careers have been like that, and it fits in very nicely to the whole arc of our course. Okay, so there's our author like that, and that's him currently down in Boston at Boston University, and this is where he did his graduate work, and you can see it's in chapter 4 because they really want to talk about this. And you don't need to know this guy's name, but his name is Howard Temin, and that's the author's graduate thesis advisor. When he was your age, he was in Madison, Wisconsin, working in that lab. And they studied these retroviruses, and they studied that Rouse sarcoma virus because it could cause cancer, like that. And again, you don't need to know on the exam, Madison, Wisconsin, 1970s, Howard Temin and Jeff Cooper, like that, right? But in the process of the discovery, this Temin figured out what it'd be like the replication cycle of the virus. That it infects the cell as RNA, then it has to get reverse transcribed into DNA, like that. And he won a Nobel Prize for that because it's a really powerful tool in the understanding of those viruses and those set of viruses that were causing cancer. So I want to take us through chapter 4 and into chapter 17, 18, 19, like that, with this kind of example. And it's a historic example, right, because it's our author's career and, of course, many other scientists' career like that. But that's the flow of it, is it starts out with this virus, which is considered a model organism. Like I say model organism because viruses aren't organisms, but it's the simplest thing. And that's a really good thing to start with because molecular biologists just do that. They just break it down to that level. They're just like, we're going to take it down, we're going to simplify it, we're going to reduce it to the simplest possible system, and then we're going to understand all the nuts and bolts of it. And it's just very much like, say, you're in a class on car mechanics, and they start out the course looking at a lawnmower, right? They're just like, so we're just going to flip this lawnmower and look at the blade and how it spins around, right? And you're like, I really want to talk about Corvettes, right? Cars are awesome. But they're like, no, we're going to get the principles out on this. And that's what the history of this field did, and that's a thread of that going through our textbook, and so we should put it in our thing as well, in our classroom as well. You don't have to scramble to write these down. I'll paste these into the message I sent today. The book in chapter 4 talks about a lot of methods, and molecular biology is about a lot of experimental methods. When I was in graduate school, my advisor taught a course, and it was a course where he never used the words like chromosome and nucleus, and instead he just described data as the evidence for what we want to talk about. It was really hard, but he's a classic molecular biologist. Where it's just one thing he would say is every model is false, right? Models are horrible, like that. Everyone is horrible, like that. The word nucleus is a model. It's horrible, like that. You know, Oh, man. And so there's a bunch of that in chapter 4. And we can pick up some of these. Many of these methods are a little bit old-fashioned now. I mean, all of them are, in the last 50 years, but some of them are a little bit old-fashioned, right? They've been supplanted with more effective, more, big-scale methods and stuff like that, All of them really are chosen to be talked about because they emphasize this is physical. The gene is physical, and it's made of DNA, and it's got the 2 parts, and we can manipulate the parts and learn about it. Like, that's physical, right? And so we don't really need to talk through all these examples, but it's important for us to appreciate that, we're reading through the book. I didn't say skip chapter 4, and so there'll be questions about, like, RNA interference, and what do we need to know about RNA interference? And I'll say, well, we didn't talk about it in class, but 2 points, like that. And really often, the 2 points are, what's it used for? If it's a method, what's it used for? And what's the feature of it? And we'll talk about that. There'll always be a feature that you would say is cute, believe it or not. Oh, that's cute. That's a cute feature. And that feature that's cute is cute because it's almost always something biological that scientists discovered. And they're, my gosh, we could use this thing in general as a method. Like, RNA interference is a natural process that scientists discovered. It's found in all eukaryotic cells, especially all plants and animals. And that's important. Important role in cancer biology and so forth. But what's cute about it is scientists realized they could exploit it. They could use it as a method to decrease the expression of any gene they want to study. So it's cute. And RNA interference is both a natural process and was discovered in things like petunia and little nematode worms. But then those scientists won a Nobel Prize because they're like, oh my gosh, you can exploit that natural process in a trick, in a method like that. And so physical, and there's a feature for each one. What do you use it for? And then what is the thing about it that makes it a tool, that made it a tool? And those are really typically the 2-point features that we want to get out of this, right? Not like write an essay about RNA interference, but like it's like boom, boom. Yeah, I know something about RNA interference. What's it used for, and what's the features of it that are, the powerful features like that. Okay? All right. So this is maybe. So I will send this out like that. Yes.
Are the features usually might be related to human use or like
Really often, yeah. If it's not direct human use, it's research in a model organism that is being done in a model organism just because it's more accessible than the human, right? So there's a lot of that, right? Where you're like, oh, okay, well, we can't really do this in human. Just because of ethical reasons like that. So we'll do it in a mouse, like that. But yeah, in general, they are methods that get done in humans or almost humans like that. Yep. Yeah. And so we'll just, I somehow kind of duplicated a couple of these slides. So let's just go, okay? So that's really common. We'll loop back on that. I have a set of slides, but just that thing of that these methods show up again because it's physical, and then they're natural processes, and then you're like, oh, that's cute, like that, and we could actually use that. We could turn that into an experimental method, into a tool, like that. And I'd say chapter 4 has a lot of that in it, like that. And again, some of them are a little bit older, and so like if somebody asks about restricted endonucleases, I would say, yep, this is what they are, and this is what they're used for. And a lot of that has been supplanted by some more modern methods like that, So, bring those questions in. That's just really valuable. And when I was in college, my classmates twice booed me, like boo, boo, like that. How does that feel, right? Because I would just raise my hand and I would just ask questions. right? Because I'm like, well, that's going to slow this down, and we're just going to reduce the amount of learning in this course to like 2 or 3 facts that I will know on the exam because they've been explained to me so many times. I'd be like, so what about this, Dr. Johnson, And people be like, shut up, Jim, you want to learn. And I'm like, I want to learn a limited amount. So I described that strategy, that student strategy that I had in college to all my classes, and no USM students seem to care. Like none of them are that much of a sort of a scrambler, like loser, like, strategic person where they're just like, what about this? And you're like, come on, let's just learn some more stuff. Okay, so molecular biology, and that's the chapter. It's a lot of this, and then we're going to drive into this example some. And I started driving into this example in the snow day lecture, and we'll loop over it because we'll hear it several times, okay? So the thing is, on our first day of class, I showed this slide, and it shows a cell. That's a cell membrane. This is the nucleus, nuclear membrane. And it shows that there's a growth factor that's going to stimulate growth. And it's outside of the cell. It could be a hormone like insulin or like human growth factor. Binds to a specific receptor. The receptor changes its conformation and gets activated in the presence of that molecule, which is a signaling molecule or a ligand. And it activates a signal transduction pathway that leads to a cell response. And there are a million different things a cell can do. Our favorite is this type of response, which is a response to a growth factor, because chapter 17 is about these pathways, and chapter 18 is about the cell cycle. So the cell cycle gets stimulated. This is not an example of regulating the cell cycle. It's a different kind of growth. It's regulating the rate of aging, but it's a good one for us to start with, because FOXO is a transcription factor, and to do its job, it needs to be in the nucleus. And this growth factor's function is to inhibit FOXO through that pathway. And that's what chapter 17 is about. It's about signaling molecules binding to receptors, activating signal transduction pathways, and its cellular response. The cellular responses fit into 2 big categories. They either affect gene expression, and those are called genomic responses, or they don't affect gene expression. Those are called non-genomic responses. Here's an example of a non-genomic response we'll learn about in chapter 17. The function that you know of insulin is this. Insulin binds to the insulin receptor on cells, especially cells like muscle cells, and a signal transduction response causes a response, and part of that response is for the cell to take up glucose from the blood. That's the familiar role of insulin. That part of that is a non-genomic response. Doesn't involve changing gene expression. We're going to talk about a lot of responses that change gene expression, just because it puts it all together. It puts all our topics together. And our favorite one is going to be the one I started talking about in the snow day video, and that is under a scab, our skin cells are growing fast to heal. And they've been stimulated by a growth factor to grow fast and heal. And that pathway is understood really well, and that's going to be our example. And so we'll talk about that some today. And that's just going to be another thing we talk about. Like, we can turn to muscles and talk about muscle differentiation and gene expression of muscle differentiation. And we picked on that example, but we could have picked on an example of brain cells and brain cell differentiation, or pancreas differentiation, right? Because it's just an example of differentiation. Then, more generally, it's an example of changing gene expression, like that. So this example of activating skin healing by increasing the cell cycle, chapter 18, of skin cells under a scab, is just an example of very general ideas, right, like that, okay? All right, so with FOXO, here's in the absence of growth factor, FOXO gets into the nucleus. and it activates gene expression. And it activates a bunch of genes. A couple hundred genes get activated in response to FOXO, and it slows down aging. Okay? So we started to talk about this, and we're going to continue to build this list, add to them, add to them, add to them. And we talked about MyoD. That's a transcription factor that's regulated just by being expressed. When it gets expressed, it binds to myogenin and activates it. Most transcription factors get expressed, but then they have a switch. They're dynamic somehow. FOXO is dynamic in two ways, right? There are other transcription factors that are regulated similarly to FOXO, or differently, right? Okay? And the one we're going to talk about that's active under a scab is a transcription factor called ELK. And ELK is there right now, but it's inactive. And when we get cut, a pathway is activated, and it's just like a series of dominoes falling, and one of the steps along the pathway is to activate the ELK transcription factor. And it gets activated by phosphorylation. So we'll have a list. There are a bunch of transcription factors that get regulated by phosphorylation. FOXO is one, really gets turned off by phosphorylation. ELK is another, it gets turned on by phosphorylation. So in our normal skin cells, ELK is there, but it's not phosphorylated, it's inactive. Under a scab, this pathway is activated, and it includes phosphorylation and activation of ELK. Okay, so that's how that fits in. And here's the pathway, and it's shown in chapter 17. But we're going to use that to just roll through chapter 4 and 9 as another example. So when you have questions about restrictor endonucleases and stuff like that in chapter 4, or gene expression in chapter 9, I'll say, well, let's use our muscle video, or let's use our skin video, or not video, but our skin example to talk about it. And so this is the pathway, and it's really 2 slides. The second slide shows that this is ELK, and ELK is going to get phosphorylated, and when it's phosphorylated, it binds, and it activates transcription. And this is a great example, partly because it's the center of chapter 17. Then, when it activates transcription, it's going to increase the cell cycle, which is chapter 18. So in chapter 18, we'll continue to talk, when we get way into 17, 18. In 18, we'll talk about ELK there because it's activating transcription of a protein called a cyclin, and a cyclin activates the cell cycle, like that, So it's just a long series of dominoes. And so we're just going to talk about these dominoes, and we'll put names on them. And the first one is that the thing that gets it started is a signaling molecule, a growth factor called PDGF that I described in this Note A video. PDGF is a great name, platelet-derived growth factor. It's from platelets. Platelets are little packets of membrane-bound material that go through our body, through our blood, and they get released at the site of a wound. And one thing they release are clotting factors. Clotting factors clot the blood, form the scab. Another thing, a different thing that platelets release is PDGF. And PDGF is released under the scab because that's where the clotting factors release, that's where PDGF get released. It's just active locally there, and its function is to stimulate growth, stimulate heating, right? When the scab falls out, your skin is pink, we got a lot of new skin cells there. They grew fast. Our skin cells are always growing slowly, right? Dead ones rub off like that. But under a scab, they grow fast. And that's really interesting to understand, and it's through this pathway. So the first thing is PDGF, then PDGF binds to a receptor, and the receptor is got a nice name, the PDGF receptor. We'll learn in chapter 17 that the PDGF is a member of we'll call them a family of receptors, and so in chapter 17 we'll talk about receptors, and there's a big category, and they're called receptor tyrosine kinases. And in humans, I think there are 58 proteins and 58 genes for those proteins that are in this category. Okay? And so one of them is the PDGF receptor, our favorite. Let's say that'll probably be our favorite receptor in our course. And these receptors are in the cell membrane. They have an extracellular domain that binds to some signaling molecule. And then an intracellular domain that is an enzyme called a tyrosine kinase enzyme. So when they get activated, they're an enzyme called a tyrosine kinase. Yep?
Was it 58 receptors?
Yeah, 58 different receptors.
Oh.
And we'll learn some other ones. One's called KIT, one's called the insulin receptor. That's a famous one. ERB, which is the EGF receptor, and there are some famous ones. Just as a little side note, this category of receptors is the one the author wants to talk about the most, because many of these are in growth pathways, and growth pathways get disrupted in cancer. So many of these receptors have stories about cancer, like KIT is mutant in many gastrointestinal stromal cancers. Most of them have a mutation in KIT, like that. So there's some really famous receptors, and many of them that are in cancer biology, and many of those are receptor tyrosine kinases, like PDGF receptor. Okay, so it's in that category. So we have a PDGF receptor, and then it's going to activate RAS, and RAS is a protein that our author studied in finishing graduate school and moving to Boston. He left Wisconsin and went to Boston to study this RAS. And RAS is a gene that's mutated in about 1/3 of all human cancers. So it's a big time oncogene, and I talked a little bit about why. And it's going to get activated. And then it's going to activate RAF, and now RAF is going to activate MEK. MEK is going to activate ERK, and ERK is going to activate ELK. And they're like dominoes. And every one of them is problematic in cancer, because we could accidentally have a mutation and have them active even without PDGF. And they're just stimulating the cell to grow and divide, even though it's not under a scab. This type of pathway is not limited to PDGF and the PDGF receptor. Many of the other receptor tyrosine kinases and even some other receptors also use this signal transduction pathway. So that's how RAS could be mutated in 1/3 of all human cancers, because it's used in many different settings with different receptors to stimulate growth, stimulate chapter 18 cell cycle, like that, okay? All right, and so that's going to be our favorite example, and it just fits together right so well. L is a transcription factor. We love transcription factors like FOXO and MyoD and myogenin and ELK. And this is the simple story of ELK. ELK is inactive, it's always there, it's inactive until it gets phosphorylated, it's active, and it stimulates chapter 18 cell cycle topics like that, okay? Okay, so it's activated by phosphorylation, and phosphorylation is done by enzymes called kinases. So that's why the PDGF receptor is called a receptor tyrosine kinase, because the internal cytoplasmic domain is an enzyme, and it's a kinase enzyme. It phosphorylates specific targets, like that. And each kinase has specific targets. So one of ERK's targets is ELK. ERK is a kinase, and it phosphorylates ELK, like that. And it's a very common way to switch a target on or off. We usually look at proteins that are targets, but kinases can have other molecules that are targets. And they're usually switching them on or off, like that. And so this is literally a chemical reaction, and it's showing the substrates are the target, like ELK, and ATP. And what it's saying here, author doesn't explain this, but ATP is an energy intermediate. So why is that showing up here? Why does it mean for this to get phosphorylated? And the answer is really satisfying. The answer is like this. This protein, like ELK, it consists, can be in two different conformations, two different shapes, inactive and active. And they're a little bit like a toggle. It can flop from one state to the other. It's a little bit like a light switch that could go in two different positions. And what you want is you want to be able to control which position it's in. You can also think of it like a teeter-totter, and you could go and the teeter-totter's wobbling like this. But you want it to be either down there or down here. You want it to be either off or on. And the ATP, the energy, is flipping it to stay that way, right? It takes a little effort to flip a light switch. That's the role of the ATP. That this light switch could be floppy. It could bounce from on, off, but you want it to be a crisp molecular switch, so you're going to incorporate a role for ATP in this reaction to make it more stably shift. It takes a little bit of energy to toggle it over, just like a teeter-totter. There's a kid over there, right? And so that could go up. It's not going to go up. But you can use the ATP energy like in your foot to just switch it over. And so all of these are designed to be evolved to be that way, where there's two possible states, and they aren't sloppy. They're kind of like a light switch. They don't just wobble back and forth. And they tend to be in one state, and the ATP energy shifts them to the other state. So that's what's going on all the time with kinases. Kinases, there may be 600 different kinase enzymes, kinase proteins in humans, something like that. You know, hundreds of different kinases. And we're learning about some of them, right? And if we just look at this, it's surprising to see this, but you see the power, usefulness of kinases. So RAF is a kinase that phosphorylates MEK and activates MEK. MEK is a kinase that phosphorylates ERK and activates ERK. And ERK is a kinase that phosphorylates ALK and activates ALK. And what's going on there is like multiple switches because we're playing with fire. You don't want to get the skin cells growing fast because you have skin cancer. So this provides for a lot of checks and balances, a lot of control, like that. And so it also provides for amplification. A little bit of PGF can really stimulate the cell cycle strongly. Yep?
Moving back to the other slide where you're showing the conformation change, like the two, like the glycogen phosphorylase. What's I understand that we're talking about kinase and that structure change, but what are the two blue balls? How do they play into this?
Yeah, the blue balls are the teeter-totter. The target is really often a protein, and the protein could be in two conformations, like that. And you could just imagine any example. Like imagine a mouth has two conformations, and one is closed, and the other is on, like that, you know? And it could be in either one, but you want to regulate it. You want to have a no eating state, an off state, and an eating state. And so the blue ball is just the protein target, and it has those two possible conformations, off or on, and the ATP kicks it over to one. Because the two conformations aren't floppy, there's an energetic barrier to kick them over, kind of like a teeter-totter, where it could be floppy if nobody's on it, but it's leaning one way because there's a kid on one end. So it has two states, but it's going to take some energy to flip over to the other state.
Are the blue balls the kinase themselves, or are they?
Well, it gets confusing when we talk about a pathway of multiple kinases, but just think of ELK and think of the fact that right now in our skin, ELK is there and it's in the inactive state.
Okay. So
But then under a scab, it gets phosphorylated and tips over to the active state.
Okay.
Yep. And ELK, it shows ELK like I'm pretty sure that when ELK gets phosphorylated, it tips to a state that binds to DNA. So when it's not phosphorylated, it's just not bound to the DNA, right? We know that, right? They've got to bind to that enhancer of the target genes. And it's got the DNA binding domain, but maybe it's closed. So it's not going to bind to the DNA and activate that gene. But then the phosphorylation causes it to just tip over to this thing, and it's now open like that and can bind. So inactive, like closed, active, open. And they're just using the blue balls as a real generic thing of just, it's not trying to say, it's showing that active being like that, where you're like, whoa, look at that thing chewing, like that, right? But we'll talk a lot about kinases. The PDF receptor is a kinase. ELK gets activated by a different kinase. And we don't have to learn that RAF, MEK, ERK stuff, some of that, right? We don't have to learn each one of those. The famous ones are PDGF, PGF receptor, RAS, and ELK, because there's something we want to say about each of those. Okay, receptor, it's a famous receptor, it's a receptor tyrosine kinase. So we'll learn something about the receptor, like that. Then RAS is used in a variety of different settings in this signal transduction type pathway. So we'll learn about RAS. And then ELK is just a transcription factor, one of 2,000 transcription factors in us, like FOXO and MyoD. And we understand how those are regulated. How do those get switched off and on? And so this is a beautiful example, because it just pulls together in just one story so much of our course that we can turn to. We can talk about, RNA interference and how it's used to study this pathway and things like that. We can just turn it. So we want to be as familiar with this as we are getting with the DNA clay model videos. So for example, here's this, like ELK. How does ELK get activated? Well, by phosphorylation, right? That's the proximate mechanism for regulating it. You could say also, well, ELK is regulated by PDGF, right? Because that's what PDGF is doing. You could flip it around and say, what's the function of PDGF? And you could say, stimulate healing of the skin, or you could say, stimulate the cell cycle, or you could just say, activate ELK. All those are, different scaling into what it's doing, right? And so we love that example, and we'll, draw that example in class and assignment, stuff like that, and talk about it a lot, okay? So here's our author, his current work, and you don't need to scramble write this down, but in chapter 17, he studies now a somewhat different signal transduction pathway. And it's a kinase that phosphorylates a target molecule that's not a protein. That's a really famous one. We'll learn about in chapter 17, like that. And we're talking about wound healing, like that. I got a couple of slides here. And it's this pathway and that story, like that. And so we add it to our list, right? And we have something to say about MyoD, right? Talking about it and, putting it into context to help us appreciate it, right? But this list is really about the switch. What makes this transcription factor dynamic? And that's because we're interested in gene expression, and the DNA is really not changing. It's these transcription factors that change, especially ones that bind to the enhancer, and they're the big switch. And so we'll have a list of these, and we'll have different contexts that we'll understand them in. Some, with more detail to help understand them. But the real focus here is like that molecular biology focus of just down at the nuts and bolts level and wondering and learning about switches. Now, transcription factors are always proteins, and so when we focus on transcription factors, we're also learning general ideas about proteins, like that. Like we're learning about phosphorylation, and RAF is not a transcription factor, right? But RAF is a kinase, and it's regulating phosphorylation, and it's involved in kinase stories. So by learning about transcription factors and how they switch, we learn a lot of protein biochemistry, right? And there's a lot of that, right? Where we talked about clay model video, but it wasn't limited to gene expression in muscle. It's a universal example, right? And we'll learn about this pathway, but there are many, many signaling pathways, you know? And it's just a good example to carry with us. And then these guys, we're learning about transcription factors, yeah, but they're proteins. And so we're learning more broadly about regulation of proteins like that, okay? All right, how are we doing? We're doing great. Because here's a figure that shows, and you don't have to try to read this, but that's the PDGF receptor and PDGF. And that's not, so what we just talked about is not the only thing that's happening in those skin cells under a scab, right? And so, you know, I know about that, and I'm teaching this, and right, I'm trying to teach it in a way to introduce the concepts so that you could add knowledge. But if we just did this, you'd just be like, I don't remember anything from college, right? But you can follow it here. There's RAS, RAF, MAC, ELK. There's other stuff too, other pathways. And they're just super famous pathways, not just in cancer biology, but of course the flip is in our normal health, and not just in growth. We'll see some of these receptors that are famous in cancer also appear to play a role in Alzheimer's, you know, in other disease states. And so it's just important to appreciate that we're focusing on examples to try to introduce the principles and get you in a position where you could spread out and learn, you know, in a more, you know, add more to it without being confused, you know, like that. But yeah, okay? And here the author also still mentions that RAF, MEC, ERK, ELK pathway, so he's working on some pathways, but he's all about this signal transduction. And why? Because it goes back to he went to grad school to study cancer. And he went to that lab in Wisconsin to study cancer. And they found stuff in the virus story in Wisconsin that led him to a lifetime of cancer research about genes like this protein RAS and the gene RAS gene, stuff like that. Okay, so let us pause and do one of these concept chats to break things up a little bit, but also focus on your notes and say, what are 3 important things that we talked about so far in class today? And we'll do that, and then we'll keep talking. And there's a sign-up sheet somewhere, and you always want to sign up if you're here. Where's the sign-up sheet? It's back there. And so make sure you sign up. Maybe I'll bring it up here. If you need to sign up, it'll be up here. But try to always get a partner. 2 people is good. If we have an uneven number of students, we need 3. But it should just be 2. And try to look around. Have masks if you think you want to put a mask on to do this. And be respectful of, you know, other people wearing masks and stuff like that. Thoughtful. But, you know, take a look around, move around, and we'll just quickly talk about 3 things that you each thought were important so far in class today. Oh, and here's somebody's pen. Yep. And we do this pretty fast, so we'll just do it for a couple more minutes, and share what you guys think, and then we'll ask a couple of people what they talked about. So we'll just do this another minute, and then we'll check it. Okay, TIVA. What is one thing that you guys talked about that you thought was important today?
The transcription regulated by FOXO.
Yep, transcription regulated by FOXO. Yep.
And how gets phosphorylated and it activates transcription. And it all starts from the signaling molecule.
Yep. Yes. Yep. Yeah. And if we just go back to that FOXO, each of these has got a story, and for the FOXO, it gets phosphorylated, and as a result, that 14-3-3 protein binds, and the 14-3-3 protein prevents it from getting into the nucleus. In the case of FOXO, I don't know if FOXO really has a really teeter-totter like shift, like that, like mouth closed, mouth open, so much as if it's just those two phosphates help create the tag for 14-3-3 to bind. Maybe, I don't know, but you can see that it looks like all you need to do is phosphorylate FOXO, and it gets to be a tag where 14-3-3 binds. And 4-3-3 is just said to be sequestered in the cytoplasm. It just cannot go into the nucleus, and so whatever it's bound to is not getting in the nucleus. Right, it's like another kid on the playground sitting on you and being like, "You're not going into school. We're skipping out." Ta-da! Right? Okay, Zechariah, are you here?
Yeah. Just something I learned just now.
Yes.
Well, there are hundreds of kinase in the cell or cytoplasm.
Yeah. So kinases are something that will come up pretty often, and that's they are enzymes, so they catalyze a chemical reaction, and the chemical reaction is going to have substrates and products, and the substrate always includes ATP, and then a product is ADP. So it's going to hydrolyze ADP, it's going to release that energy, and then it does that. As it's doing that, it takes that phosphate and covalently bonds it to the other substrate. So the products are ADP and a phosphorylated product, and that phosphorylated product is the thing we're interested in, that is somehow switching it, and it could be switching it on or off. In the case of elk, it switches it effectively on. In the case of FOXO, it's really turning it off. So we always have to look at the targets to see, well, is the phosphorylation activating them or inactivating them? Because it could go either way. It's specific, but like that. And then, yeah, Zofia, what's one thing you guys talked about?
We talked about how some proteins are turned on by phosphorylation while others are turned off. There are 58 different receptor tyrosine kinases.
Yep.
And what I thought was really cool is that DNA viruses such as Epstein-Barr, HPV are associated with 38% of all virus-associated cancers.
Yep. Yeah, and that last thing is something that we'll learn about in chapter 20. So, we're going to go 4, 9, 17, 18, 19, 20. And 20 is really what I think it's called cancer, like that. And it's the last chapter in our book. And there we'll learn about that. For now, we mentioned, we'll bring in some other viruses for two reasons. Other viruses for two reasons, being that molecular biologists used to study and still study viruses because they just like to reduce things to the simplest thing, like a lawnmower, or even just the lawnmower's wheel. And you're like, we want to study cars, and they're like, look at this lawnmower wheel. And you're like, yeah, really brutally reductionistic like that. Then second reason that we study other viruses is that some of them play a role, like HPV is that viruses that cause genital warts that can become cancerous. It doesn't directly cause cancer, but it leads to a precancerous state of overproliferation and benign growth, like a wart. The virus that we're going to focus on are these category retroviruses, and then just a small number of them that are like variants that happen to have picked up a problem and can directly cause cancer, okay, like that. And we're going to talk, a few more minutes like that today. Do people have questions, issues about anything they want to ask about? Yep.
What's the like glycogen phosphorylase glycogen and then there's one phosphate in that action? Like what's
Yeah, so see what's going on here is it's saying there's a pathway with like dominoes falling, right? And that first word epinephrine used to be called adrenaline. And so that's this response of like, oh, the tiger walks into the room, and all of us go, oh, we got to run away, right? So it's that rush that adrenaline causes, right? People talk about adrenaline or epinephrine. It's going to cause this tremendous rush, right? And throughout our body, really fast. And what's happening, it shows, is that epinephrine would be, it's a hormone, it's a signaling molecule, and it's binding to receptors, and then the arrows are that signal transduction pathway. And in this case, that adrenaline rush is not a genomic response in the target cell, not gene expression. That would not be fast enough, right? We don't have time to transcribe a gene and have that somehow help us get out away from the tiger, right? So what it does is more than this, but this is one really, really good example. So this glycogen phosphorylase is an enzyme. It's inactive, and that pathway is going to lead to its phosphorylation in activating it. So it's one of these toggles, and it goes from like an inactive enzyme, so like a closed mouth, to an active enzyme, kicks over, toggles over to this active form, and what it does is it breaks down glycogen. And glycogen is a long polymer of glucose that's stored in our cells. And what it's doing is it's very rapidly catalyzing the breakdown of glycogen to glucose, and it's literally a sugar rush. So if you eat a candy bar, you get a sugar rush. This is a sugar rush, but not from eating it. The sugar is stored as a polymer, waiting and hidden in every one of our cells, especially muscle cells. And the epinephrine activates the glycogen phosphorylase, and it just rapidly breaks glycogen down and releases huge amounts of glucose right inside the cell. Like no need to chew and digest. It's just the sugar's there. It's just been trapped in glycogen, and it's just a whoosh like that. And that's the effect, right? Everybody goes, oh, adrenaline. Even saying it, you get kind of jumpy like that, right? Because you just are like, yeah. And what's happening is that, and other things too, but that's just a really classic one. And that would be an example, again, of a non-genomic cellular response. Most of the time, we're going to talk about genomic ones because we love transcription vectors, we love gene expression like that. They're just such satisfying stories like that. But there's a really good example where you're not going to wait around for a transcription factor to do its job. You're just going to release a huge amount of sugar like that. Yep. Yeah, and that's from I guess it's coming up in chapter 2 because it's about metabolism. Chapter 2 and 3 are about metabolism, and we're skipping over those like that. But this comes up in chapter 17 when it's talking about some pathways. Yeah.
So, in essence, epinephrine's able to like trigger this faster than transcription factor. Is that kind of what you're saying?
Well, it's just yes, and just to talk about this idea that it's a hormone, and all hormones are signaling molecules. They're going to bind to receptors, and that's going to cause a cell response. And almost all the time, we'll talk about cell responses that involve changes in gene expression, genomic responses. There are many, many, many cell responses that don't have changes in gene expression. This is one of them. Not all those happen really fast, but this is a cute one because it happens really fast. Another really good example of a non-genomic response is all neurotransmitters. Those happen really fast. And those are signaling molecules, and all neurotransmitters are signaling molecules that bind to receptors on target cells, nerve cells, and the nerve cell makes a cellular response. In that case, in microseconds, right? So just changing the resting potential across the cell membrane, and changing the diffusion of ions across the cell membranes like that. So, there are lots and lots of examples of these, and we're introducing them, and we're introducing the ones we love the most, and that's because we're molecular biologists, and we love to talk about changes in gene expression like that. Yep. Other questions people have about stuff right now? Yep.
Something's phosphorylase.
Yeah, so so for every kinase activity, there really needs to be an opposite activity. So if we're turning ELK on, we need to be able to turn it off. And the enzymes that reverse kinase activities are called phosphatases. And so let me just erase this.
Is that why when you're lysing samples to prep for PCR, you use phosphatase inhibitors?
Sorry, what?
Is that why when you are lysing cells collected to run a PCR, you use phosphatase inhibitors?
So when you're lysing cells and you use a phosphatase inhibitor,
So the proteins don't get degraded that were at the time of cell collection?
Are you using a phosphatase inhibitor or are you using a protease inhibitor? Yeah, okay. So I would say, like, I don't know in that situation, like, why you're using that phosphatase inhibitor.
I don't either.
Yeah.
But
Yeah, I mean, I'll think about it because I used them a lot, but I'm just not thinking about what the explanation is.
sorry,
Yeah.
I didn't mean to
Yeah,
derail.
but anyway, so here's a chemical reaction, right? And it's catalyzed by an enzyme that's called a kinase, right? A kinase is an enzyme that catalyzes this kinase reaction, right? And we're going to have a phosphate added. And there's enzymes that catalyze the exact opposite reaction. So we have hundreds of these enzymes in our cells and bodies, and they're called phosphatases. So they do the opposite reaction. And I don't think a phosphatase requires ATP, because I think that, like, it's like this when you step on the teeter-totter. I think it's hard for it to flop over to the phosphorylated state, but it's like easier to remove that, and when you remove it, it will flop back. I guess, yeah, the gas pedal is sort of springy like that. But I mean, that's a detail, but just to say, I don't think ATP is a substrate for phosphatases. I just don't think they need it. They just are removing it and it naturally springs back like that. Yep. Other questions people have about stuff? And we'll talk a lot about kinases. We won't talk very much about the phosphatases just because they're reversing that, and that always has to be available like that. We'll talk about a couple as we go through our course. Yeah. And we're in good shape. Let me just show you a slide so we're I got to hand back your thing. So let me hand back your quizzes. And so the thing is, I made this answer key video, and I'll just have some stuff marked off. You know, that's wrong. And I tend to just say that's wrong, that's wrong, that's wrong, and it's like 2 points for each question like that. So I've marked those, and you know, I'll send the link today for the answer key video, and you want to look through that. Even if you got them all right, you want to look through that just to see, because I don't talk about students' names, but I talk about how students answer the questions, right? And a lot of times I say, well, you know, this was the good answer. Students gave this answer. Most of the students gave this good answer. And if you didn't have the good answer, well, it was 2 points off, like that, okay? So, this is the first one of these, and it's just a quiz, right? So it's not worth a lot, but we want to focus not so much on the points, but on the learning, right? All of a sudden, we're learning. So here's TIBA, and when you can head out, and LA, and Brooklyn, and Amal, and Jeremy, and Eva, and Catherine, and Broadway, and Samantha, and Sophia's coming up here. All right, so like I said, the answer key video for this is just 10 minutes long. So, you know, everybody could take a look at that, and that'll be great.